Next up, we have Joey Matt, who's we've worked with before, worked with him, and he's going to talk about investigating some of the genetic issues with triploid mortality. This will bring you, that goes, the down arrow goes forward.
Hi everyone, happy to be here today. Today I'm going to be talking about unusual mortality events, what I'll be referring to as triploid mortality, which has been responsible for significant crop loss on farms in Virginia. And as the title suggests, I'm going to be talking about a project looking into the genetic basis, or what genes have to do with these mortality events. So first, some background. The first major reports of these unusual mortalities were received by VIMS in 2014 when 14 farms reported. For most farms, these mortality events lasted about 4 weeks and killed over half of the affected stock. All the farms had in common were the time of year this occurred, which was late spring, and the type of oysters that were affected, which were near market-sized triploids. This led to these being called as triploid mortality, which is how I'm going to be referring to these mortality events throughout the rest of the talk. So since then, there's been at least 8 reports of matching criteria, and at least 4 within the last year. And here's a map of the Chesapeake Bay indicating some of the major regions where VIMS has received reports. And what makes triploid mortality unusual is that it's not associated with the typical factors of oyster mortality, like large swings in salinity, extreme salinities or extreme temperatures, or evidence of disease pressure from MSX or dermo. So what do we know about triploid mortality? I'm just part of the research effort at VIMS looking into triploid mortality, so I'm not going to present all the findings, just some of the major ones. Number 1, a strong side effect. We know this from the most recent study in triploid mortality, where we deployed the same diploid and triploid crosses to various sites in the Chesapeake and saw triploid mortality at only 1 site. Number 2, multiple types of triploids are affected, or strains. We saw this from the same experiments where genetically different triploid crosses suffered triploid mortality. Number 3, diploids may fare well. From the same experiment, we saw Debbie diploids had high survival during a triploid mortality event. Okay, so there's also been reports of mortality events in triploid stocks on farms in Alabama and Louisiana, suggesting these similar triploid mortality events may be occurring in the northern Gulf of Mexico. In 2018, there was an especially similar report to what we found in the Chesapeake, where farmer reported high mortality in late spring in their triploids and low mortality in their diploids. Wadsworth et al. From the Auburn University Shellfish Lab followed up on these reports with a field trial examining survival in diploid and triploid crosses in sites in the northern Gulf of Mexico, and found mortality similar to what we recently found in the Chesapeake. A potential key difference, however, between the findings in the Chesapeake and that in the Gulf is the different environments these mortality events are occurring in. At the same time this mortality happened, Wadsworth et al. Also saw a large shift in salinity, which has been absent in our observations of triploid mortality in the Chesapeake Bay. Okay, so findings are good, but solutions are better. So considering the findings we've just made, we just talked about, let's go over the solutions for farmers. Okay, number 1, there's a strong side effect. Okay, so if you're in a site with triploid mortality, just move your farm to where it isn't. Thank you, I'll take my award for that. Okay, no, but getting more serious now, another option, we saw diploids perform well. Maybe you could grow diploids. Well, then you're missing out on all the advantages of growing triploids. And number 3, what I expect most farmers are doing is they're growing more with an expected loss. The point in showing all this is to say that there's really not a great solution right now. And what would be a great solution is if triploids could grow, or farmers could grow triploids that are resistant to triploid mortality, which brings me to the point of this project, is to see if this was possible. Can we breed for triploids that are resistant to these triploid mortality events? In order to know if we can do this, we need to know the genetic basis of triploid mortality. Triploid mortality can be thought of as a trait, just like these other traits here on the left. And when I'm talking about genetic basis, what I'm saying is, to what extent are genes controlling the trait? The expression of a trait could be controlled by genes or not. So take, for example, here's a population of oysters. The trait we're looking at is survival, and in generation 1, we see many oysters die. We take those survivors, we breed them, and in generation 2, we see higher survival. This increase in survival, assuming the environment is same from generation 1 to generation 2, can be attributed to the good genes in the survivors from generation 1. So we say there's evidence of genetic control for survival in this case. We can take it a step further. We don't have to think all or none, and we can think about it on a spectrum. One end, no genetic control. On the other end, total genetic control. This is what is referred to as heritability for a trait. So if we were to map the traits I have here on the left by their heritabilities, you may see something like this. The point is, the higher the heritability for the trait, the higher the genetic control, and the more we can do about it with selective breeding. Okay, so how do we measure heritability? At the Aquaculture Genetics and Breeding Technology Center at VIMS, we do this by testing families. In breeding lingo, family is just a group of brothers and sisters. So in this slide, the circle is a female, the square is the male, and the triangles are families, groups of brothers and sisters. So we do this because it allows us to track the genes. Each family can be thought of as a set of genes, and each family can also be thought of as a different set of genes. So now we can associate differences in the trait to differences in the genes. So take an example here. We're looking at mortality, and we measure mortality in all these families, and we don't see much variation among the families. That suggests the genes don't really matter, and it suggests this trait has low heritability. On the other case, we have high variation among the families, which suggests the genes matter a great deal, and there's high heritability for this trait. Okay, so moving on into the experimental design of this project, ABC made 20 triploid families in 2017. Each triploid family was made with a different tetraploid male, and each tetraploid male was spawned to a common pool of diploids. We also wanted to include tetraploids and diploids in this field trial for comparison, and we did that by including oysters spawned in 2017 from diploid high salinity lines, diploid low salinity lines, and tetraploid lines. These oysters were deployed to 3 sites in the Chesapeake Bay. 1, the York River, Virginia, which is a high salinity site. 2, the Chesapeake River in Maryland, which is a low salinity site. And the bay side of the eastern shore of Virginia, which is also a high salinity site. At the York River and Chesapeake, oysters were deployed in longline baskets. And in the eastern shore site, they were deployed in bottom cages. All the triploid families and tetraploid lines were deployed to each site, but only the diploid high salinity lines were deployed to the high salinity sites, and only the diploid low salinity lines were deployed to the low salinity sites. We replicated. We had 3 replicates per family or line per site, so that meant we had 81 total bags or baskets per site. Okay, last, we measured these oysters. A measurement comprised of counting all the live oysters in the basket, taking a sample of 15, and measuring shape, size, and condition. Here's a timeline. We deployed the oysters in 2018. First measurement was the following fall, and then we measured 3 times in 2019, spring, summer, and fall. I just want to point out we expected to see triploid mortality between the spring and summer of 2019. Okay, here's a picture of the ABC team, which did so much work for this project, and the reason we got such good data. Speaking of data, here's some. Here you're looking at cumulative survival from the time these oysters were deployed to the end of the experiment. And here in gray are the triploid families, in white are the diploid lines, and in black are the tetraploid lines. And at the bottom here you see average monthly salinities corresponding to those months on the x-axis. And the first thing I want to point out is the tetraploids had the lowest survival. This isn't surprising, as Jess from ABC tends to say, tetraploids like to die. But other than that, what we see is relatively little variation in survival among the triploid families. Okay, moving on, same format, different site. Now we're looking at the low salinity site, the Chesapeake River in Maryland. Again, we see the tetraploids had the lowest survival. But what we see different here is much more variation in survival among the triploid families. But what we don't see is we don't see an acute mortality event in late spring. Instead, what we see is a more gradual mortality ever since the oysters were deployed. And this mortality curve makes sense considering the especially low salinity we saw at this site during this time period. Which really suggests is if we captured variation in a different trait, something like low salinity tolerance. There's more to that story that I can't go into now, so I'm just going to leave it at that. Going into our last site here, our bay side of Eastern Shore, I'm just showing part of the data here to show how similar this looked like to the York River just leading into the spring. But between the spring and summer, we saw an acute mortality event that matches our criteria for triploid mortality. Looking a little closer at the data for this event, this is just survival between May and July at the bay side of the Eastern Shore site. The first thing we see is that the diploids had high survival, which again suggests that diploids may do well during these triploid mortality events. And the other thing we see is a lot of variation among our triploid families, from 100% survival to 60%. And remember, this suggests a high variation among families, suggests the genes matter, and suggests there's heritability for this trait. And we are able to quantify this heritability through quantitative analyses, and we measured this heritability at 0.43, which in the animal breeding world is moderate to high and suggests we may be able to do something about triploid mortality through selective breeding. Okay, so to summarize, one, we found some evidence similar to what we found before, a strong side effect. We only saw triploid mortality at one site. Again, during that triploid mortality event, we saw the diploids survived well. Two, we saw high variation in survival among the triploid families during the triploid mortality event, from 100% to 60%. And three, we were able to measure a moderate heritability in this trait of triploid mortality, which suggests through selective breeding we may be able to have substantial impact on this trait in future generations. Okay, so look, some caveats and looking ahead. One, this data is site limited. We were only able to measure triploid mortality at one site, and in order to better understand this trait, we need to expand these type of progeny tests to more sites and regions. Two, this data is family limited. We were able to make estimates based on 20 families, but to get more robust estimates for heritability and other genetic parameters, we need to increase our data set with more families. And ABC has started this by making another set of triploid families in 2019. And last, a little bit looking ahead, there is some more data coming out from this project looking into why certain triploid families were susceptible and others weren't. And we're going to do this by looking at a high sample size. We have 45 samples from each triploid family just before the triploid mortality event. So we're going to be able to look at their gonad and their biochemical composition to look for something physiologically distinct in those triploid families that were susceptible. Last, I want to say thank you to the ABC team. Thank you to my advisor, Stan Allen, and thank you all for your time.
Thank you, Joey. Any questions for Joey? We'll switch over. Well, Beth walks that over.
The stocking density was 120 oysters per bag or basket.
So is there a way to, it seems like that heritability ought to be coming from the tetraploids. Is there a way to test your tetraploids before you breed them?
Well, that's part of the breeding process. So now we know from the tetraploids we used which ones may be more or less susceptible to produce triploids that have this trait. So then we can know we don't want to use tetraploids that are closely related to the parents of those the parents of those triploids that died. So that's all part of the breeding and the pedigree-based breeding.